Assalamu alaikum everyone. In the previous lecture, we started discussing the regulation of the trip operon and how the trip operon is turned off if tryptophan is present inside the bacteria, and some of that tryptophan will bind to a protein called the trip repressor, activating the repressor so that the repressor or the active form of this repressor will bind to the operator, preventing the RNA polymerase from initiating transcription. If there is no tryptophan, the trip repressor that is encoded by the TRPR gene, because of the absence of tryptophan, it will remain inactive. It cannot bind to the TRP operator, so the RNA polymerase will be free and will be able to initiate transcription. Okay, so this is one example of an operon in bacteria. Today, inshallah, we're going to learn about another example operon. So in general, there are two types of operons. We have repressible operons and we have inducible operons. This is the TRP operon is an example of repressible operon. Repressible means al-muthabbat. It can be repressed once the repressor is activated in the presence of tryptophan. So the normal or the default situation or the default state is to have transcription from the TRP operon unless the repressor becomes active after the binding of the tryptophan, and now the active repressor can repress transcription or expression from this operon. Okay. So there are two types of operons. We have repressible operons, we have inducible operons. So repressible operons, they are usually on, they are usually transcribed, unless a repressor or active repressor binds and turns transcription off. Inducible, they operate in the opposite, in an opposite fashion. So these inducible operons are usually off, but when a molecule called the inducer inactivate the repressor, transcription will be turned on. Okay, so the TRP operon is an example of a repressible operon. Now, these repressible and inducible promoters are example of negative gene regulation. Negative gene regulation means these are the genes that could be turned, or expression of these genes could be turned off by a repressor. Okay, so we have two types of gene regulation. We have negative gene regulation and we have positive gene regulation. So in negative gene regulation, we have two kinds. We have inducible operons, we have also another type, repressible operons. Two types. And these operons are, or these inducible repressible operons, like we have the trip operon and we have the lac operon, which we will be discussing today, inshallah. Both are examples of negative gene regulation. It's called negative gene regulation because operons, this is very important, because operons are switched off or turned off by the active form of the repressor. In positive gene regulation, we have a, let's say, a regulatory protein switch transcription on. So a protein will switch transcription on. So here we're talking about a repressor that will switch the operon transcription of the operon off in positive gene regulation, and we will learn some examples. So far we have discussed the trip operon as an example of a repressible operon operating basically in a negative gene regulation fashion. The lac operon, which is an example of an inducible operon, this operon can function in both fashions as a negative for negative gene regulation and positive gene regulation. So an example of another example of a negative gene regulation operon is the lac operon, but at the same time, as we will see today, the lac operon also could operate in this positive gene regulation fashion. So lac operon is also another example of one example of positive gene regulation. So let's now discuss how the lac operon functions, or how it can be regulated. So the lac operon consists of, just like the trip operon, genes, operator, and ribosome binding site. There are 3 genes, lac Z, lac Y, and lac A. So lac Z encodes an enzyme called beta-galactosidase. This enzyme normally converts or splits lactose into galactose plus glucose. So this is the normal function of this enzyme. We have also the lac Y, , So the lac Y encodes an enzyme called permease. This enzyme is important for the transport of lactose from outside bacterial cells into the inside. And lastly, this enzyme, well, it's written here. This enzyme is trans-acetylase. These are enzymes. This is an enzyme that is, its function is still, undecisive or unknown, let's say. So we're talking about enzymes. Let's say this is bacteria, E. coli. This is a bacteria. And bacteria, like E. coli, has in its genome the lac operon. So this is the lac operon. The lac operon encodes an enzyme called the permease and the beta-galactosidase. So this is the permease, and this is the beta-galactosidase. If there is lactose, so the bacteria, these two enzymes, or the lac operon, allows the bacteria to uptake lactose from outside, and then this lactose, using the enzyme beta-galactosidase, it will be converted into glucose and galactose. And both could be used to make more compounds or more substances or more organic molecules inside the bacteria, but also to make, so this is chromosomal DNA, and to use these two molecules to produce energy inside by respiration inside the bacteria. Okay. So the permease is a membrane protein that allows the lactose to move from outside the cell into the inside. And once the lactose inside, the beta-galactosidase enzyme can convert this lactose into glucose and galactose, and these could be utilized for different purposes inside the bacteria. Now, the default state or the normal state of this operon is to be off, so that its transcription is turned off. So we have a regulatory gene called LAC-I. This LAC-I encodes an active repressor, and this active repressor binds to the operator, and there's no transcription or no RNA, and the operon is turned off. This is the default state or the normal state. Because you have to understand, bacteria, and just like many human cells, they prefer to produce energy from glucose first. If there is no glucose and there is lactose, they will utilize lactose to produce energy or to synthesize other organic molecules. But the priority is to use glucose first. So typically there will be, if there is a lot of glucose, there is no need to turn on the lac operon because glucose is available and the bacteria can easily convert the glucose into other organic molecules or maybe use it to make energy or ATP. But if there is no glucose, okay, so if the glucose concentration is very low, and this is very important, and there is lactose, then the lac operon expression will be turned on. How? So in the absence of lactose, okay, this, and there is glucose, the operon will be turned off. But if there's lactose, and it's very important, and the concentration of glucose is low, what will happen is that some of the glucose, some of the lactose will be converted into a molecule called allolactose. Okay? So some of the glucose, lactose, will be converted into allolactose, and this allolactose binds to the repressor, which is the product of the LAC-I gene, and makes it inactive LAC repressor. And this inactive LAC repressor cannot bind to the operator, so transcription will be turned on from this operon, leading to an mRNA, one mRNA molecule. Okay? And this mRNA molecule can be used to make the three enzymes important for lactose utilization. So this is an example of inducible operon. Why it's called inducible? Because normally it is turned off and it will need as a molecule called inducer to induce its expression or to turn it on. Okay? This is inducible promoter. The trip operon is a repressible promoter because normally it is turned on. This is the normal situation. Transcription from this operon is normally, or the default state of this operon is to be turned on, unless a repressor binds to the operator because of the high concentration of tryptophan and repress the expression from this, or transcription from this operon. Okay? So we have two types of operons, inducible, repressible operons, and both are example of negative gene regulation. And I would like to emphasize more about the differences between inducible and repressible promoters. So let's compare inducible versus repressible promoters. Operons. So in inducible promoters, we have an inducer binds to the repressor. Let me go back here. An inducer called allolactose, which is basically is converted from lactose. So when lactose is present in large quantities, some of that lactose is converted into allolactose, and now this allolactose will behave as an inducer. This inducer, it's called inducer because it will allow the initiation of transcription, or it will allow the induction of transcription by inactivating the repressor, by preventing it from binding to the operator. In the repressible operon, we have the co-repressor, a molecule called co-repressor binds to the repressor. Okay. Another difference between these two operons is that in the inducible operon, under normal conditions, like here, come on, in the inducible operon, under normal conditions, the repressor is active. This is the normal condition. In the repressible operons, under normal conditions, the repressor is inactive. So it is synthesized, it is expressed in its inactive form. Once it is produced by translating this mRNA, it is normally inactive. Here in the lac operon, which is an inducible operon, it is translated directly into an active form. So it is already active. So we could write here, under normal conditions, the repressor is active. Here, repressor under normal conditions is inactive. It will be activated by the binding of the co-repressor. So in the repressor operon, the repressor is inactive, and in order to become active, it needs a co-repressor. Let me go back again here. The co-repressor is actually the tryptophan. Another difference here, the inducer, for example, the lac operon, the allolactose, turns on the transcription. Here, the co-repressor, if the co-repressor binds to the repressor, the co-repressor turns off the transcription. Typically, inducible promoters are important for, usually used for anabolic pathways. Anabolic pathways, and I will discuss this point in a moment. And repressible promoters usually are employed in catabolic pathways. So a pathway is simply a number of consecutive chemical reactions that are important for the biosynthesis or the degradation of molecules. Anabolic means anabolic pathways. They are important for let's say so anabolic and catabolic. So remember that metabolism, عمليات الحيض, consists of two major branches or two major types of reactions. We have catabolism, and we have anabolism. So here, oh sorry, this should be the opposite. Anabolic, this is catabolic. Catabolism, hadm, bina, anabolism. So remember, in the lac operon, the lactose is split or catabolized into glucose and galactose. So this is catabolism, عملية هدم. In the trip operon, we have several enzymes. Let me go back to this slide. Several enzymes that are important for the synthesis or anabolism of tryptophan. Okay. So this is the difference between catabolic pathways. Catabolic pathways are the pathways or the chemical reactions that are important for the degradation or the breakdown of molecules. Anabolic is the pathways involves biochemical reactions that are important for the synthesis of molecules. Typically, the inducible promoters, they are turned on in the presence of its substrate. This one is turned off. Or maybe its product. Let me write it. So its product or end product exerts feedback inhibition to shut off the path, the expression from the operon. So turned on in the presence of its substrate. Remember that in the lac operon, the presence of its substrate, the lactose, turn on the operon. Okay? So this operon, the expression of this operon is turned on, okay, in the presence of the lactose, which is its substrate, because these enzymes, they use this enzyme, and this enzyme, they use lactose as their substrate. In terms of the trip operon, for example, its end product. So this is the trip operon. The trip operon genes, they are important for encoding these enzymes. These enzymes, they synthesize tryptophan. Tryptophan, this is a metabolic pathway. This is a metabolic or anapolic pathway. It's the same thing. Metabolic is a metabolic anapolic pathway because it is used to synthesize a molecule. And this is the pathway end product, منتج النهائي. And as you can see, the end product can exert feedback inhibition on the pathway to shut off the synthesis of more of the end product if the concentration of the end product is high. And finally, an example of an inducible promoter is the lac operon. This is an example operon. And here we have the trip operon is an example of a repressible operon. Okay? So next we're gonna discuss how mutations might affect the lac operon and how these mutations can affect the expression from the lac operon. So we're gonna talk about lac operon mutations and see how different mutations affecting different regions within the lac operon might have different effects. So I'm gonna make a table and we're gonna divide this table into three columns. I'm gonna write here the mutation. And I'm gonna write also the function. The function affected by this mutation. And lastly, the phenotype that might result from this mutation. Now, before we discuss these mutations, you have to understand that if you look at the LAG-Z operon, we have several genes. We have LAG-Z, LAG-I, LAG-A, we have LAG-I. Also here we have what we call LAG-P, which is not a gene, but it's a symbol to indicate that this is the operon, the part of the promoter where the RNA polymerase binds. So LAG-P, P for promoter. And we could call this DNA region as LAG-O, so P for promoter or polymerase binding site, let's say, and O for operator. Of course, this piece of DNA does not encode any protein, but the LAG-Z encodes a protein, and the LAG is for the lac operon, and A and Y, they encode proteins. The lac I also encodes a protein, but these two, they do not encode proteins. They are just DNA sequences. Okay? So we could say that we have a phenotype or a mutation called LAC-Z mutation. So if a mutation is introduced into the LAC-Z, we have LAC-Z mutation. Normally, we write the gene. So these genes, as you can see, or these regions of genes or genotypes, this is a genotype, LAC-Z mutation. In bacteria, most genes are written using 4 letters. The first 3 letters are lowercase, and the last one is uppercase. If we're talking about the LAC-Z protein, it should be written like this. So LAC-Z mutation, or if we speak about gene, sometimes we might use LAC-Z minus for a mutation and LAC-Z plus for the wild type or normal gene. This is a mutant gene which has a mutation, LAC-Z minus. Okay? So LAC-Y minus is a mutant gene, for example, and LAC-Y plus, or maybe just LAC-Y. So sometimes we use the minus plus sign, the minus to indicate a mutant gene. Sometimes we use the plus or not to indicate a normal or what we call it wild type gene. Okay. And sometimes we use minus superscript or maybe other letters. Sometimes we use other letters to indicate a type of mutation. We will discuss the different types of mutations and the meaning of the superscripts that might be added to indicate a type of mutation or a particular type of mutation. Okay? All right, so let me just draw a bacteria. And we have the bacterial chromosome, or maybe we have the bacterial chromosome, but then we have the lac operon. I'm gonna just amplify the size of the lac operon. And then we have the lac I. So I'm gonna use just letters. This is A, lac A. This is lac Y. This is lac Z. This is O. This is P. This is I. So lac I gene here is indicated by Y. Lac P, which indicates the polymerase binding site, is just P. And then we have lac O equals O here, and lac Z, Z, Y, A, just to make things more simple. Imagine this is an E. coli cell. Now, E. coli cells, they are considered haploid cells. Do you know what's the meaning of haploid? Haploid. What does it mean that E. coli are, or E. coli is haploid organism? Anyone? What's the meaning of haploid? If you don't know haploid, what's the meaning of diploid? Anyone? Haploid, diploid. شو يعني أحادي? What's the meaning of أحادي? In terms of genetics, genetic terms. So haploid organisms, they have only one copy of each gene, or a single set of chromosomes. Diploid organisms, like humans are diploid because we have two sets of chromosomes. One set you inherit from your father, one set you inherit from your mother. So you have 23 chromosomes from your father and 23 chromosomes from your mother, which means we have, for most genes, we have two copies. One copy you inherit from your father, one copy you inherit from your mother. E. coli or bacteria, they have only one copy of each gene because they have only one set of chromosomes. Of course, bacteria, they have only one chromosome, so they have one copy of each gene or one allele of each gene. But we can, in the lab, introduce, let's say on a plasmid, you don't know what is a plasmid, but we can introduce another copy of genes into bacteria. We could do that easily, like this. We can introduce it into the bacteria. So now the bacteria, just the lac operon. Imagine if we could introduce into the bacteria just another copy of the lac operon, like this. So normally bacteria, they have just one copy of all genes, including the genes of the lac operon, but we can insert from outside another copy. So we have two copies. So now the E. coli is haploid for the lac operon, but now it is called not diploid, but now it is called merodiploid for the lac, just the lac operon. So we did not insert any genes, so any other genes. So this is just the chromosomal DNA of the bacteria, just one chromosome. It's a circular chromosome. And we artificially, بشكل صناعي, we introduce another copy. So now the bacteria is considered merodiploid. Merodiploid means just partially diploid. We do not call it diploid because we did not provide another copy for all other genes. We just only provided or introduced another copy only of the lac operon. Okay, so now we have two copies of the lac operon. This copy that is normally found on the chromosomal DNA, and this is the other copy that we introduced artificially into the bacteria. Again, it's not called diploid because we have only two copies of the lac operon. For the rest of the genes in this bacteria, all these genes, all the other genes have only exist inside this bacteria in a single copy. Why do I need to insert another copy of the lac operon? I will tell you later, but for now, it will be used to determine the dominance and recessiveness relationship between the mutations of each of these loci. So each of these genes or regions in the DNA could be called genetic loci. These are genetic loci. Each one of them can be called locus. So this is the genetic locus of lac I. This is a genetic locus called lac B. This is a genetic locus called O, lac O. This is a genetic locus called lac Z, which happens to be also a gene. Now, let's go back to this table. Let's look at the different mutations. So the first mutation affects the enzyme, the gene lac Z. In this case, the function that is affected, let me go back to the slide. Notice that lac Z encodes the beta galactosidase. So the function that is affected is the beta galactosidase enzyme activity. So if there is a mutation that affects the lac Z gene, there will be no active beta galactosidase. Now the phenotype is called lac minus. Notice that this is to indicate a mutation in the gene. This is just the phenotype, the general phenotype. So the bacteria, now the phenotype of the whole bacteria, if there's a mutation here, okay? So this star means mutation. So now this is Z minus, okay? So the phenotype is called lac minus. This mutation is recessive, and I will show you how it is recessive and what's the meaning that it is recessive. Okay, maybe I should write it like recessive. So the mutation is recessive, not the bacteria. We're talking about the mutation, this mutation or this allele. Now, we have two alleles, lac Z plus allele, which is the wild type allele. And we have another allele called lac Z minus, which is the mutant allele. Just like when you learned about recessive and dominant alleles in the chapter about Mendel work, for example, in peace plants, we have dominant capital letter, we have small letter recessive alleles. Here, we do not use capital and small letter, we use just positive, negative, or maybe letters, superscript letters or symbols, like positive, negative. How did we know? But remember, when you talk about dominance and recessiveness, we typically discuss dominance and recessiveness in the context of diploid organisms, because these organisms, they have two alleles or two copies of the same gene. Bacteria are haploid. We cannot apply the dominance recessiveness relationships to haploid organisms because they have only one copy of each gene or one allele of each gene. This is why we used or we introduced another copy to determine if this mutation or this mutant allele is dominant or recessive with the respect of another allele or another normal allele that is found inside the cell. So, because now we have two alleles, two different alleles, this is a wild type allele, okay? So I could say, I could just put, this is Z plus, and this is Z minus, or like Z minus, like Z minus, like Z plus. It's the same thing. So now we have two alleles. We have two copies of the same gene. We have two alleles of the same gene. One of them is a mutant, the other is normal. Now we can determine the relationship between them. We can determine which one is dominant, which one is recessive. The lac Z is recessive. Do you know why? The lac Z minus mutant allele is recessive to the lac Z plus. Do you know why? how did I know that the lag Z minus allele is recessive? And how did I know that the lag Z plus is the lag Z, the wild type lag Z on the other copy of the operon is dominant? Think about it. What's the function of the lag Z? To produce what? Beta galactosidase. Okay. What's the function affected if there's a mutation lag Z minus? The beta galactosidase active. So if we add to this bacteria lactose, and now the lactose will enter the cell, what will happen to the lactose? The lactose inside the cell, if there is a beta galactosidase enzyme, okay, it will be able to convert lactose into glucose plus galactose, right? But we need the enzyme beta galactosidase. Now the enzyme beta galactosidase could come from here, from this gene, because this is a gene that encodes the beta galactosidase, and this is another gene that encodes the beta galactosidase. So we have two genes that are called the lag Z genes. But this one is normal, which will give the beta galactosidase. This one is not normal. It has a mutation, so it will not be able to produce active beta galactosidase, and there will be no active beta galactosidase. So the only active beta galactosidase will come from this wild type copy of the gene, and this wild type copy of the gene is enough to convert the lactose into glucose and galactose, which means the whole phenotype of this bacteria, the final phenotype of this bacteria is lac plus, which means it can split lactose into glucose and galactose because of this dominant allele. So the phenotype of this allele is dominant over this one. The phenotype of this allele is the production of active beta galactosidase that can convert lactose into glucose and galactose. The phenotype of this allele is the absence of any activity and the absence of the inability to convert lactose into glucose and galactose. But because of this one, this enzyme or this gene is enough, it will produce enough enzymes to convert lactose into glucose and galactose if lactose is present. Okay, to make this bacteria lac plus, which means it can break down lactose into gal plus glu, galactose and galactose. This is what is meant by lac plus. Lac minus is the opposite. It cannot. Okay? This is why the phenotype of this allele is dominant over this allele. So the bacteria can utilize lactose. Okay? Is it clear? Yes. Good. The phenotype also, we could add something called, this is a trans-acting mutation. So it's a recessive mutation. It's a trans-acting mutation. Trans. So let me discuss. We have we can divide DNA sequences into two major types. We have, come on, shoot this on. We have trans-acting and we have cis-acting. So trans-acting DNA sequences or and cis. These are DNA sequences, but they can regulate. So trans-acting, we have example trans-acting sequences. These are factors that affect the expression of distant genes. Cis-acting, they regulate the transcription or expression of nearby genes. Come on. بعلق بالمرة. So here we're talking about cis-acting, we call them elements, genetic elements, عوامل جينية, trans-acting elements. So example of trans-acting elements, we learned about transcription factors. Transcription factors are proteins encoded by other genes that can regulate genes that are far away. Transcription factors, we learned about the repressors in the lac operon repressors, for example. Repressor proteins. We have also like the RNA polymerase also, it's a protein that can diffuse or move from one gene to another. So for example, if we go back here, notice that the LAC-I gene encodes a protein, and this protein can go bind into a distant gene and regulate the expression of another gene. So this is an example of trans-acting. Do you know what's the meaning of trans and cis? Do you know what's the meaning of trans? Trans means, what does it mean in English? Trans, anyone? Means across, عابر, or beyond. Sorry, beyond. عابر بالعربي. Because here we have a trans protein that can cross or go or diffuse into another location within the gene. So this is a gene encoding a trans-acting. So this is a trans-acting element because it can produce a protein, diffusible protein. Diffusible means it can move, can diffuse, يتحرك into another location and regulate the expression of another DNA sequence. Okay? Cis, on the other hand, cis means, I don't know what's the definition of cis. And, cis, how can we define cis? Cis means on the same, let's say, on the same structure or the same molecule. So trans, we're talking about two different sides, maybe, or cis on the same side, okay, on the same molecule. Let me So example of cis-acting elements, we have promoters, because promoters, there are sequences that regulate the expression of genes downstream them, promoters, okay, operators also. So here we have a promoter that basically drive or control the expression of other genes on the same DNA molecule or nearby genes. This operator is also important for determining whether these genes will be transcribed or not. Okay? So these two sequences, promoters, operators, activators, silencers, they are examples of cis-acting elements. Now, cis-acting elements, they affect the expression of the allele on the same chromosome. So if we're talking about here, these two cis-acting, they can affect the transcription only of these alleles on the same chromosome. And these two can affect the transcription of these alleles only. These two cannot affect the transcription of these genes. And these two cannot affect the transcription of these two. This is what is meant by on the same gene. On the other hand, these trans factors affect the transcription, the expression of both alleles equally. All right. What does it mean? The LAC-I produces the LAC-I repressor protein. And the LAC-I repressor protein can both bind here and bind here which means the LAC-I repressor protein can affect the transcription of these genes on this allele and this allele, or this allele and this allele equally. Okay, this is what is meant by affecting the expression of both alleles equally.